안녕하세요. Thank you for tuning in this morning, and thanks for the nice introduction. So today, we're going to talk about some of the genome editing tools and workflows for cell engineering that we've been developing at Thermo Fisher. So as an agenda, I'd like to start with an introduction on genome engineering and some of the history of genome engineering as to how we have arrived at where we are today. We're going to talk about the different genome editing tools that are available. We have a case study on creating disease models from human iPS cells. And we'll finish on talking about the newest tools to the market, which are CRISPR-Cas9-based gene editing tools. And we'll talk about how these tools can be used for multiplex gene editing. And we'll finish today talking about complete workflow solutions that we provide from Thermo Fisher. So if we talk about cell engineering in terms of synthetic biology, we approach this field thinking of it as a bit of an engineering challenge. And we look at this as a cell engineering cycle as an understand, build, engineer, and test cycle, as we would looking at a normal engineering problem. So as we look at this in terms of the mammalian cell, what we would initially like to do is to understand as much about our mammalian cell platform as possible. The information that we can get from these cells comes first from next generation sequencing, so we can understand the genome sequence and identify and profile the different types of mutations that may be present in the cell line that we're interested in. We can also overlay this information with profiling of the transcriptome, so look at the messages that are being produced in the cell to try to understand the cellular metabolism at the time. So from this information, we can hopefully create hypotheses to be able to allow us to design our editing tools. As we get to the building of the genetic element editing tool, we use a lot of synthetic DNA synthesis that allows us, as we'll show you, that allows us to build the actual tools that go in and edit the DNA. It also allows us to build the donor DNA that we use to repair or replace some genetic elements within the cell. As we move to engineer the cells, we look at delivering these tools. So most of the tools are encoded by nucleic acids. So we work very closely with our delivery genome transfection and delivery groups in Carlsbad to create optimized solutions for delivering these specific editing tools. We'll have some data on that at the top. Certainly, we've got expertise, and we're working more and more on using chromosomal integration systems, using different integrases to be able to insert up to very large elements into the genome at defined locations. We are also working on not only cleaving the DNA, but also modulating the cell metabolism using transcription factor-like tools for regulating gene expression and also doing regulating epigenetic effects in chromosome remodeling. And I'll talk a little bit about genomic circuit design. We're actually heading in that direction. And that's basically what I think of as applying synthetic biology mechanisms or expertise into cell engineering. So once we engineer our cell, we'd like to analyze it again to understand how what we've done to our cells. So we would go back to doing the next generation sequencing and also look at transcriptome profiling again to understand if we've knocked out or modulated a particular gene in one pathway, if that had an effect on any other pathways within the cell. Now this stage represents a big challenge for us because we can generate a lot of data, a significant amount of data on these cells. The challenge here is when we try to take these large data sets and analyze them to be able to get a clear indication of how we've affected the cell's metabolism. So we have a lot of work going on with our bioinformatics team to try to develop tools and solutions that will allow the biologists to be able to understand these multi-layered data sets to understand what we've done to our cell. So eventually, as we iterate around this cycle, we'll arrive at a point where we've edited the cell or engineered the cell to what we've desired, and then we can use these modified cells for our experiments. So really, what is genome editing? It's actually very simple. What we like to do is take our DNA and insert create a double-stranded break. And to that allows us to add DNA at this location. So we can insert new genes, we can insert reporters. We can also replace existing DNA and either repair or create mutations. So as small as single-base mutations, we can repair and replace those. We can also remove some DNA as well. And so we'll go into a lot more detail about using these editing tools and using the cell's repair machinery to create knockout, easily create knockout in genes. And what kind of applications can we use for this? So basically, we can study gene function by doing a gene knockout, and that will create what's called a loss of function study. So we can try to get an understanding of how this particular gene's activity relates to the entire cell metabolism by removing that function from the cell and seeing how the cell responds. We can also, like I said, insert mutations into the cell, single base or multiple base mutations that would change the coding sequence of a gene or potentially change how a promoter functions. And we can see, try to understand what the effect that has. We can target transgene in addition for a heritable modification. We can move toward creating an engineered tissue to produce a novel function. And we can also put in endogenous gene labels so that if a particular gene is activated during differentiation or during cell metabolism, we can then identify when that gene is activated. Our lab focuses more right now on stem cell and creation of tissue disease models, and we'll show you that. We're working in collaboration with some researchers on editing and engineering plant tissues to create more robust plants, crops for maybe disease resistance or drought resistance or salt resistance, or also working to create better, new and better biofuel platforms as well. Eventually, as we get good at this, I think we'll be able to support the gene therapy and cell therapy market. We're also working in collaboration to use these new editing tools to create new and better disease models in small animals. And what's not out here is we have a significant amount of effort going on in our lab and also with collaborators to engineer cells for better bioproduction platforms. So creation of biopharmaceuticals, mostly in hamster CHO cells or potentially in human cells as well. So engineering a cell has lots of different applications. So how have we gotten to where we are today? If we look down here at the bottom left of this chart, is basically, I would say 20 years ago or so when we started getting interested in actual cell engineering. Back in those days, our view of cell engineering was basically inserting a gene into the cell and trying to express it. This was done just by transfection of the cell with a DNA expression plasmid. with an antibiotic selection marker. We would then add antibiotic to the culture and select a population of cells that had our expression construct randomly inserted into the genome somewhere. We had no idea where it would go. We would screen lots and lots of colonies to try to identify cells that had the best expression of our gene. We were at the mercy of the cell as to where these things integrated. We did not have an understanding of how the genome would respond. Oftentimes, the gene expression would silence after time. So we had no control, really, no precision of where these things integrated. If we wanted to do a mutational study, we would then mutagenize the cell line by either exposing it to high doses of radiation or potentially a chemical mutagen, and then again we would screen and screen and screen to find a phenotype that was of interest, and try to understand what mutation led to that phenotype. So it's a lot of work and a lot of hoping, I think, basically, in those days. So not long after that, RNA interference and antisense RNA started to become popular. And what this allowed us to do was to design molecules, RNA molecules, that would knock down a particular transcript of a gene. It would not specifically knock it completely out, but you would get a decrease in expression. And this was a really good tool, and it moved the field ahead very quickly. But we learned quickly that actually these tools were not that specific. So you might knock down the gene that you're looking for, but you oftentimes would knock down a family of transcripts that might give you a phenotype that's maybe not so specific as the one that you're hoping for. So the RNAi knockdown screening requires oftentimes follow-up of screening to look at those gene expressions in orthogonal ways. Meanwhile, there was groups working mostly in mouse cells at the time to do targeted integration of genetic elements via homologous recombination. This was a large step forward in the field. It required creating targeting plasmids that had oftentimes very large flanking homology regions, so up to 3 to 7 to 10 kilobases on either side of the element that we want to insert into the gene. This is not always very specific and efficient, so it took, again, a lot of screening to be able to isolate the clone, the properly edited, engineered clone that we're looking for. So several years ago, we're introducing genome editing tools. So there were a tool called meganucleases. Around the same time, there's a tool here, zinc finger technology. So basically, the inventors of this technology, if you will, they took a known transcription factor binding domain, a zinc finger binding domain, and were able to link enzymatic activity to those domains and program those domains to deliver that activity to somewhere in the chromosome. So this was a huge step forward. Allowed us to now create double-strand breaks in areas of chromosome that we're interested in. These tools were not terribly easy to engineer. It would take a lot of some screening of libraries of zinc finger combinations to be able to direct them to the appropriate location in the cell. But at the time, it was definitely a very, very useful tool. Once you arrive at an optimized zinc finger tool, then those elements are actually very useful, and they remain useful today. In 2009, Jens Bach's group in Germany and others identified the TAL effector technology for use in mammalian cells for editing. Now, these are proteins from plant pathogen, bacterial pathogen organisms, Xanthomonas and Ralstonia. Long story short, I'll go into some more detail later. These tools are very easily engineered to bind specifically at a chromosomal location. And so we can get very high specificity of directing these tools to specific locations of the genome. And we can, as with the zinc finger technology, we can use these as a vehicle to deliver enzymatic activity to specific addresses in the genome. And about a year and a half ago, the latest entrant in the field of programmable DNA binding proteins and editing tools came along. This is another prokaryotic system that's been now converted for use in mammalian cells, and this includes CRISPR-Cas technology. And we'll go into a lot of detail. So the summary of this slide is basically we've had a very, very rapid evolution of tools, especially in the last 6 to 8 years, to create solutions that allows the researcher to create specific double-strand breaks in the genome, to which we can use other tools to be able to engineer themselves. So where do we think we're going to go next? So if we look at this now we're down in the lower left corner again today, and all the things that I'm showing here on the slide are actually being currently worked on. And so we think this is at least partially the future of the field. So we'll start with CRISPR-based knockout libraries. So I mentioned RNAi and what a breakthrough that was several years ago, but there was some shortcomings. Now using this CRISPR tool, which we'll show you more detail on, now we can package that CRISPR tool in other delivery vehicles, perhaps lentivirus, perhaps other things, to be able to go and do genome-wide or gene set-wide screens to do actual knockout of gene activity. So in contrast to a partial knockdown of the transcript that might not be specific, CRISPR promises to allow us to be able to go in and specifically knock out of the expression of a gene at the promoter. So what we're seeing initially and what we're anticipating is that this tool will allow us to be able to do these loss-of-function screens in cells and be able to do it much more specifically than with RNAi. So the phenotypes that we can get from these screens we feel are going to be much more reliable and robust. Secondly, we look at the emergence of these editing tools and how efficient they are. We're looking at the next real pain point in the workflow, and that would be actually increasing the efficiency of insertion of our donor DNA or our repair into the genome at these double-stranded breaks. The whole idea is to make the workflow as efficient as possible to allow the researcher to get from their original hypothesis and idea to the actual cell line that they can do their experiment with. So the more efficient you make every step of the process, the fewer clones, cell clones one needs to screen at the end to get to the desired cell. So we and others are doing a lot of work in this area too. Secondly is an area that we and others are just really getting started with. And this is really where synthetic biology is joining with cell engineering to a certain extent. Now we can look at, take our learnings from synthetic biology over the last few years and research that's been done mostly in prokaryotic cells to create logic circuits similar to the type of electrical circuits we would see in electrical engineering. We can create logic gates that would be a NOT gate or an AND gate or a NOR gate that would be able to measure stimulus in the cell's environment in different ways. And if these stimuli provide a particular profile that we've programmed into our logic gate, then that would trigger some response in the cell. It could be a reporter gene. It could be some other response that would activate an internal pathway within the cell to allow it to respond to these stimuli. These are very early days. I call it creation of smart cells, and I really truly believe that this is part of the future of cell engineering and synthetic biology. And at the end of the arrow, which is certainly not the end of at the end of this journey, if we can learn all these editing tools and create the efficiencies and get the expert at creating these logic circuits, and we can combine this with tissue culture technologies that will allow more efficient 3D cell culturing and creation of multicellular tissue-like what we call organoids. If we can combine this with our other gene editing and synthetic biology technology, we really believe that we will be able to derive more physiologically relevant tissue culture models or disease models that will actually allow us to do more efficient drug screening in the context of tissue. So currently drug screening is generally done on cell lines that are maybe transformed cell lines that are growing on plastic surfaces. And that is about as far away from the situation in the body as we can get. So our goal is to work with the field and develop internally new tissue culture platforms that will allow us to be able to allow our partners to be able to screen better drugs and be able to get better drugs out to the market sooner. So we're talking about genome editing tools a little more specifically. As mentioned, we've had an evolution or a revolution in genome editing tool development. And essentially, it's very simple. The evolution and revolution has actually been the ability to deliver a nuclease activity to a specific location in the genome. So we're basically delivering the scissors. What happens when we do that delivery is we create a double-stranded break, generally, in the genome. And when we do that, several things can happen. There's two pathways the cell can go down to be able to respond to this double-stranded break. The first is a repair pathway called the non-homologous end-joining pathway, where the cell wants to seal that double-stranded break. The good news for cell engineering is that the cells are generally not very specific when they do that. So there's often a deletion or a small insertion of DNA at these double-stranded breaks. And what that allows us to do is if we create that break within a protein coding sequence, open reading frame of the gene, and that repair occurs, then more often than not, it's going to create a frameshift mutation, which would more often than not introduce a stop codon in the gene. So by creating these double-stranded breaks and relying on the sloppiness of the cell's repair machinery, it's a very easy way to create a knockout of a gene. And so the field has, in the evolution of these gene editing tools, there's been a lot of interest in creating knockouts. It's currently so far. Now we're seeing, as the field is becoming more comfortable with these technologies and with creating knockouts, there's more interest in actually going in and editing the gene. And that would be, instead of just creating a lesion to create a knockout, now we want to go in and change one base or 5 bases in terms of DNA. So to do that, we want to encourage the cell to join a different pathway. And so in addition to adding our editing tool, we would add a piece of donor DNA that would then be inserted into this double-stranded break. And so the pathway that is used to do that is termed homology-directed repair. So this is a separate biological pathway within the cell. And this is much more efficient than the homologous recombination pathway that we've used previously in mice that required a lot of homology regions. And essentially, the HDR pathway really only needs between 200 and 500 bases of homology on either end of the double-stranded break. So that allows us not only to make the plasmids easier to create our inserts, but we can actually synthesize these donor DNAs and use double-stranded DNA, or actually use single-stranded oligos to do this repair very efficiently. So that allows a lot of versatility in creating our donor plasmids. And again, I mentioned that we and others are working on making this pathway more efficient. So I alluded to the fact that we've had this revolution in genome editing that's been really led by the development of these tools. So if you look at the graph on the left, you look at the number of publications for genome editing, we can see now an inflection point when the entry of GFNs into the market. And as you see, things are evolving maybe logarithmically, because we have the tau effectors come out in 2009, and we see an inflection point on publications. And not long after that, we now see CRISPR just a couple of years ago, and we expect another inflection point there. So these tools, the efficiency of creating this double-stranded break in the genome, has really enabled the field to move faster and allow more researchers to be able to enter this field to engineer their cells. Without exception, these tools are very robust. We focus in human cell line mostly, and in plants through our collaborators, but you can see examples in these publications of these tools being used in a broad array of host systems, including flies and worms and mice and fish and cows and monkeys and pigs. So basically, I don't know if there's many or any organisms that these have been tried that it definitely don't work. And again, the focus here for us is mammalian cells. We'd like to do work on generating stem cell disease models, and we're also working in bioproduction. So this slide just gives a quick snapshot of the tools that have been evolving in order. So first there were the meganucleases and the zinc fingers. As you can see from the cartoon, the way the zinc fingers work is that we have two zinc finger nuclease molecules binding around a cut site. Each of them bring with them a monomer domain of BOC1 nuclease. And if the zinc fingers bind appropriately around the cut site, these domains dimerize and create a double-stranded break. Similarly, with tau effectors, we bind these in opposing regions of the DNA. They bring along with them their BOC1 nuclease and cleave the DNA. Now, the tau effectors were very interesting because these were found to be consisting of repeated protein domain structure. And each repeat domain is found to be responsible for binding one specific nucleotide. And so once Jens Bach's group was able to crack that code, if you will, in 2009, that allowed us to relatively easily recode these proteins to be able to bind any address we like in the genome. And again, where we can create double-stranded breaks. In contrast, the CRISPR-Cas system basically consists of a nuclease that has no DNA specificity until it complexes with a small guide RNA. So this is now an RNA-directed nuclease that then efficiently locates itself to the genome and creates the double-stranded break. So before we get into too much detail about CRISPR-Cas, I want to give an example of a project we did to engineer a disease model in human iPS cells. And we started this project in late 2012. And so this was kind of before CRISPR-Cas really was proven to work in mammalian cells. So we've done this project with tau effectors. Everything you see here can and is being done using CRISPR, but this is just a great example of our first pass to be able to use the power of somatic cell reprogramming and genome editing to create patient-specific disease models for doing basic research and potentially drug screening. So we were in a collaboration with the Parkinson's Institute in the U.S. And our stem cell group. And the project basically is the Parkinson's Institute is provided us with skin biopsies from certain Parkinson's patients, and then we had taken the rest of the workflow to create these disease models, and then the Parkinson's Institute would then go do biochemistry and some screening. So just a brief background on Parkinson's disease. It's a neurodegenerative disorder. It's generally a late onset disease affecting the elderly. This is the second most common neurodegenerative disease behind Alzheimer's. Essentially, the symptoms lead eventually to severe dementia. And basically, what's known is it's marked by a loss of midbrain dopaminergic neurons. Okay. So there's a wide span of symptoms that I mentioned that the mechanism of which are not exactly understood. So our goal is to try to get a better understanding of this mechanism. So Parkinson's Institute sent us several skin biopsies from these patients that are indicated in the lower left panel. We have controls. We have 2 Parkinson's patients with their associated specific known mutations that are linked to Parkinson's, either in the LRRK2 kinase gene or in the GBA gene. We also had a couple interesting Parkinson's-like disease patients, and this is called multiple system atrophy, and we'll go into more detail about that. So basically, the workflow is we got these skin biopsies, we grew out the fibroblasts in culture. We then reprogrammed them to iPS cells using our cytotune reprogramming system. We then resequenced those cells to identify, to confirm that they did indeed have these specific mutations in LRRK2 and GBA, at least. As I'll show you, we tested the karyotype before and after we engineered them with the tau effectors. We also checked with our potency markers, and then the idea then is to then use our tools to differentiate these cells, the engineered and non-engineered cells, into neural stem cells, and then eventually into neurons and glia and astrocytes. So again, the idea is to start with our iPS cells from our patients, either with or without mutations. Then we edit some. In this case, we're either knocking out a gene, or entirely knocking out a gene, or repairing 2 mutations. And now we have 2 populations that are match set, and they are identical in the genetic and the epigenetic background, and they only differ from each other by the exact mutations that we've induced in the cell. So now we can differentiate these eventually into neurons in this case, and we now have a match set disease model where we can compare directly the effect of these specific mutations in identical isogenic backgrounds. It's very important. So again, we use tau effectors here, but again, this is essentially only a vehicle to deliver a nuclease to do a double-stranded break. Tau effectors, as I mentioned, they're programmable DNA binding molecules that come from this plant pathogen, Xanthomonas or Ralstonia. In nature, the bacterium sits down on a leaf of the plant and injects this protein directly into the cell. The protein carries with it a transcriptional activator, which it takes and binds to specific promoters in the plant genome, activates these specific genes, and makes the cell more receptive to infection. So what we've done here is then we've removed the activation domain and we've fused this nuclease monomer domain to the tau effector. We can program in the address that we'd like to deliver the nuclease to, and then we can assemble a TAL effector, or a designer TAL effector, if you will, that then delivers these tools specifically to the genome. They bind in forward and reverse orientation, and then the block 1 monomers dimerize and then create our double-stranded break. And that allows us to either create an indel to create a knockout mutation, or if we add our donor DNA down, we can add essentially the workflow is such as this. So we were interested in doing this to use all the tools that we've developed across then Life Technologies and now the Life Sciences Group in Thermo Fisher, that basically to identify and show that we have tools to address the entire workflow. And what we'd like to do also, this is a kind of an initial project to prove out our ability to do this type of services for our partners and customers. So we have everything from the IPS culture. I mentioned the cytotune kit to rapidly reprogram cells. We have in this case, we used our neon electroporation system to deliver the TAL effector plasmids. We synthesized our donor sequences at our GeneArt facility. We have our EVOS microscopes, which we use to identify colonies and do colony picking. I show you our mismatch cleavage assay, which we use to evaluate the efficiency of the cutting of our tools. We use our TACMAN assay to identify appropriately edited clones, and our ION PGM to sequence the loci. And then when we look at expansion and characterization, of course, we have lots of tools, media and stains and other types of microscopic systems. So the workflow is as follows. We've now engineered our cells, our IPS cells, and we plate them at a relatively low density. And if you've worked with IPS or ES cells, you know they grow in colonies. So then we manually pick the colonies and we split them in two. Half the colony we kept in a plate for expansion, and half the colony we used for analysis. So the analysis workflow consists of isolating the genomic DNA from these cells. We do a PCR reaction where we amplify around the intended locus for engineering. And then we can use that product either in a TACMAN SNP genotyping assay, where we can use TACMAN probes that will specifically identify either the wild type allele or our edited allele. And that we can do that in a 96-well plate format to screen clones to quickly identify the ones that are either heterozygous or homozygously edited. What we generally do first is to amplify these down and to do a GeneArt cleavage detection assay to get a quick idea of how efficiently we're cutting at our locus. And then of the selected clones, we can take that same PCR product and put that into PGM sequencing to specifically sequence that locus to understand what the actual base sequence of that mutation is. So briefly on the GeneArt genomic cleavage detection kit, we've developed this system to be optimized for our workflows. It's an add assay in the sense that you don't need to isolate or purify any step here. It's basically you just add more reagents. You start with your colonies. We have our lysis and extraction reagents to isolate the genomic DNA. This is all then compatible with the PCR amplification buffers and the PCR enzymes. So we amplify around the locus that we're interested in, and in a percentage of that population that will contain our edit or our indel, as seen in the red spot. Now we can melt this pool and let it re-anneal, in which case the mismatched or the indel will re-anneal with a wild type, generally strand, creating, now this creates a mismatch bubble. And to this, we can add our mismatch detection endonuclease enzyme that recognizes these bubbles specifically and cuts. And so then we have a pool of PCR products, some which are full length, which don't have an indel, some that are cut, indicating they contain this lesion. And it's an easy matter then just running this pool out on an agarose gel and then calculating the amount of cleaved product in relation to the total amount of PCR product. And that gives us a very easy and fast identification of how efficient our tool is working. So let's get into some specifics. We talked about multiple system apathy. This is a Parkinson's-like disease. There's no known genetic risk factors, no mutations known. Basically, it's only characterized by these protein deposits called Lewy bodies. And what we wanted to do is to knock out the gene in this protein deposit, the term alpha-synuclein. So I'll just take you through that. We did use tau to knock out the gene. We screened colonies, and colony number 4 has this nice cleavage event, which suggested we got a good cleavage. We then sequenced to confirm we did have a knockout. Once we cured the colonies, we had about, the second subcloning, we had about 50% edited, suggesting we had knocked out one of the alleles. We then, of course, checked the karyotype. So now we have a heterozygous knockout. Make sure the karyotype is normal. We checked all the pluripotency markers, make sure they're still acting like stem cells. We then also did a Western blot to show that we're getting a decreased amount of protein and confirmed that by immunohistochemistry. So to get the second allele, we just went around and did the whole process again. And then we applied these cells to our TACMAN phenotyping assay. So again, this is a 2-color screen. So the one TACMAN probe would recognize the wild-type allele. And let's say that's the red probe, and if the cells come up all red, that means that's wild-type positive, homozygous wild-type. If the cells come up all yellow, in this case, that means that they're at the double knockout. And if they're a mixture of red and yellow, that means they're probably still heterozygous. So we quickly identified clones that were indicated that they were homozygously knocked out. We then sequenced them again and showed that in the first allele we had a 2 base pair deletion, which would cause a frame shift. In the second allele we had an 8 base pair deletion. Both of them led to stop codons, ensuring that we have a knockout in those cells. We then checked Western blot and immunohistochemistry and showed that we now have, when the double knockout in the far right lane, we have no detectable alpha-synuclein protein. So now we have that match set. We've sent that back to the Parkinson's Institute, and they're doing biochemistry on that. So a little bit more, we wanted to go in and edit some genes. So we have these 2 mutations specific for linked to Parkinson's disease. In this case, there's the LRRK2 leucine-rich kinase. Basically, this mutation from G to S at this position leads to higher kinase activity, and it's linked to Parkinson's. The mechanism is unknown. And then also there's this mutation, the GBA mutation that's linked to Gaucher's disease. This N to S mutation is linked to Gaucher's, which also brings along with it a bifold risk of developing Parkinson's. These are both heterozygous mutations, so they're only mutations on one allele. So we wanted to go in and repair these mutations and to create these disease sets. So basically, we did that. We created our tau effectors to bind and cleave in here right about where the mutation is. We created our donor DNA, which has about 500 base pairs of homology either side, and this is a linear piece of double-stranded DNA. We then did our screen. We cut and did our insertion, screened about 140 colonies and got 2 clones that looked like they were repaired. And in the first subcloning, we were able to prove that we had 100% now repaired wild-type allele. So now we've repaired that LARC2 mutation in that cell line. And then we checked it using the standard tools that I've already showed you. Secondly, for GBA, this was a little bit different because it turns out that GBA has a pseudogene that we didn't want to edit. Fortunately, there is a gap in the sequence in the pseudogene that only appears in the parent gene. It happens to be downstream from the intended mutation fix. We were able to design tau effectors to cut specifically in that gap, so it specifically cut the pseudogene, specifically cut the parent gene. And we added our fix, and basically we're able to fix this mutation way down upstream from where we actually made the cut. In this case, we got 1 out of 50 clones that had this fix, and we ran it through our screen and showed that it was repaired. So now we have 3 disease model sets that we can use for basic research and for screening. We can look at IPS levels, we can look at neural stem cells, neurons, and glial. So we can now test these paired mutations in the same backgrounds for how they go through development during this disease, and then also in the adult neurons and glia to understand if there's a different pharmacology. Just briefly, we tested the LARC2 mutation in a couple assays to get an idea whether they're behaving as expected. This is an experiment looking at glucose sensitivity. In the first control, we're looking at the number of cells in the assay, so we'll have about the same number of cells. In the disease-relevant phenotype in the middle, we're looking at glucose sensitivity. So in the mutant, if you remove glucose, then the cells don't survive well. So as you can see in the red bars, in the mutant, once we remove the glucose, those cells started to look unhealthy. But as we repaired that gene back to wild type, now we're showing that those cells, the repaired cells, are behaving like the wild type cells as expected. Similarly, if we use a LARC2 inhibitor in the absence of glucose, in the mutant cell lines, the mutant cell lines are sensitive to glucose and they're not healthy. The inhibitor will bring that back. In the case of the edited cells, now they act like the wild type. So this is the first indication that we're looking at to understand whether these cells are behaving as expect, and basically they are. And then now the Parkinson's Institute, along as well as us, are doing more types of biochemistry and screening. So we have a lot of excitement about these cells. We'll be able to use them for basic research, and eventually, hopefully, we're doing some relevant small molecule screening for maybe some therapy. This is basically the study that I've already gone through. Now I'm going to actually finish up talking about really the newest tool on the market for doing genome editing. If you remember, I talked about basically these tools are very important as vehicles for delivering nuclease activity to the genome. Cas9 is a system that is, in nature, is responsible for an adaptive immunity response in bacteria upon an infection of phage. So when the phage particle infects the population of bacteria, the surviving cells actually record the actual sequence of the phage genome and keep them at what's called a CRISPR locus. Each of these colors is actually a recording of a particular phage's genome. And these are then processed into small RNAs. These are called CRISPR RNAs. And they reside in the cell. The next time that cell population is infected by one of these phages, now the cell can use these RNAs to recognize the phage genome in complexes with the Cas9 protein and actively destroys that phage genome. That's protecting the population from further infection. So we've used that, we and others have used that to as a genome editing tool, basically. So in this case, the most widely used protein, the Cas9 protein, is from strep pyogenes. And the system basically is this protein, the Cas9, which contains the nuclease domain, again, has no DNA specificity until it's complex with this guide RNA. So if you remember the last slide, we had these CRISPR RNAs that led to our adaptive immunity. This part of the guide RNA is responsible for locating this complex to the DNA. This upstream piece, termed the tracer RNA, in nature is a separate piece of RNA that helps the complex form. In practice, the field has fused these into one piece of RNA, and now that's termed the guide RNA. But the business end of it is down here. This is what we code specifically then to direct this complex to a location in the genome. What that does, that brings these nuclease domains in contact with the DNA and allows them to cleave. We can knock out these nuclease domains one at a time to create nicks, or both to create what's called a dead mutant to allow us to deliver other types of enzymes to the DNA without cleaving it. One other piece of information that's important is that there is one requirement for this complex to be able to recognize a target under the DNA, and that's what's called the PAM sequence. These PAM sequences are actually related to the species of Cas9 that we use, and this is not coded in the guide RNA. These PAM sequences are recognized by a protein domain within the Cas9 protein. And it's thought that as the protein complex is scanning down the DNA, it's a PAM sequence. It slows down long enough to allow this domain in the protein to actually insert itself into the DNA and cause some single-stranded character, thus potentially allowing the guide RNA to associate with its sequence, and then it creates a conformational chain, which then brings the nuclease domains in contact with the DNA to create the double-stranded break. We have launched certain products into this field, and so this is our journey through making the workflow easier. We initially launched 2 plasmid versions into the space, basically encoding Cas9 protein and having a cloning construct to be able for us to clone templates for our guide RNA. And recently we've launched a solution that has, instead of a plasmid-based system, we now have purified Cas9 mRNA. And then our guide RNA templates can come in the form of linear double-stranded DNA strings. And that would either be a string with a U6 promoter allowing expression and transcription of the guide RNA within the mammalian cell, or we have a string with a T7 promoter which will allow us to create purified guide RNAs in vitro, and then to have a complete RNA solution. And all these get us to what we need as an efficient, specific double-stranded break to allow us to do our engineering. So secondly, we've developed a design tool. I know there's several design tools out currently in the space. We have ours that we've developed in-house. That allows us to identify favored CRISPR sequences, guide RNA sequences. So basically what it allows us to do, and eventually this will be available, will allow the customers or us to put in a gene ID or a set of gene IDs into our algorithm. The algorithm then pulls out that gene and the exon structure and finds the first constitutive exon in the gene. It designs a set of guide RNAs according to the rules that we give it, including the PAM sequence. It then blasts each of those guide RNAs against the genome and gives it a score as to what we feel is the goodness of that. And I'll go into a little bit more about that. And then in the end, we get a table of desired or favored guide RNA target sequences, and each guide RNA target sequence will have a table along with it that has potential off-target sites. And so we're actually creating this table for the entire human genome right now, 23,000 genes, and we actually should have that completed within the next few weeks. And so then that'll allow us a rapidly screenable database to identify ideal guide sequences for knocking on genes. So quickly, our scoring algorithm takes our desired guide sequence, screens the genome. If there's an identical sequence somewhere in the genome, it just throws it out. It gets a very low score. And then depending on where the mismatches are, it gets a higher and higher score. So for instance, if there are mismatches in the distal region of the guide sequence, then it gets a pretty bad score because the chances of the Cas9 still binding and cleaving are relatively good. As mismatches move in closer to the PAM sequence in what's known as the C region, then the chances of it actually binding and cleaving go down. And so that would get a higher score, and then these might show up. These might get a little bit higher score and end up up on our list. So basically, we did launch vectors, and like I said, we had two versions, one with the fluorescent protein tag along with one with the membrane tag. They're very useful, and I think they're doing very well in the field. But we found that they do still have some drawbacks. In a plasmid, you have to have a promoter to drive the expression of the Cas9 protein as well as the guide RNA. That brings with it some constraints as far as the different cell types. Some promoters do that better in some cells, and some do worse. Also, the payload size. So these are large plasmids, and it lends to us a restriction of the ability to put in multiple plasmids into a cell, and really decreases our ability to do multiplexing. So that's why we've arrived recently at this solution of creating purified messenger RNA for Cas9, and then also having a solution of linear DNAs for our guide RNAs. So this eliminates the promoter constraint for Cas9 expression. The payload size drops down. It allows us to optimize dosages between the Cas9 mRNA and the guide RNAs, which then makes it amenable to multiplexing. So the workflow is as this. Basically, the customer will submit a target sequence or a list of sequences. We will then design guide RNAs, and we will then create them either as a U6 string or as a T7 string. That we then deliver the mRNA along with our U6, and then transfect and analyze as we've discussed. So basically, the first thing we wanted to do in this experiment is to see how well we could multiplex with this. So briefly, we're looking at one experiment where we checked several different combinations of guide RNAs. We're looking at 3 loci in the cell: HPRT, AAVS1, EMX1. The purple bars are what was our standard, which is the all-in-one vector. And then as we move down, we have different combinations of these linear U6 guide RNAs. So I'll just point your attention down to the columns 12 and 19, which is the blue, on my screen anyway, blue bars. Basically, these are where we've added 3 different guide RNAs plus the strings mRNA. And we can show we still have very efficient cleavage in these cells. So slight decrease in efficiency, but not that much. So this was our first example, really, in our lab of being able to efficiently multiplex. These are 3 guide RNAs. We've actually gone up to 4 and 5 guide RNAs, and still we're able to get efficient cleavage with these clones. So secondly, with the T7, that allows us to create a full RNA solution. And basically, we can take these T7 strings, put them into our modified omega short script in vitro transcription reaction. And that's a 2-hour reaction on a heat block, and then it's a quick cleanup column. And now we have purified guide RNA that we can combine with our Cas9 mRNA. And what that allows us to do is to be able to multiplex, but it also has other benefits that we're seeing as far as efficiency. So I want you to remember back when we were talking about Tau effectors and iPS cells. And our cleavage efficiencies there were a little over 5%, maybe 7%. And we were very happy with that. And now when we take this Cas9 mRNA and the IVT guide RNA, and we formulate that with our newest lipid configuration, which will then be released in Asia in October, now we're seeing cleavage efficiencies almost to 50%. So we think this is a huge advance. We're getting very efficient cleavage, which means when we go back and redo these disease models in iPS cells, instead of screening 100 or 200 colonies, we might get away with screening maybe 20 colonies or maybe 50 colonies. So significantly less work to get to the clone that we're interested in. As I mentioned, it was our goal. A little bit more detail on that. If we compare the plasmid DNA, even with lipofectamine 3000, which is our newest plasmid DNA, or transfection DNA, we were getting almost 9% cleavage, and actually we were pretty pleased with that until we tried our mRNA solution with our newest lipid. In this experiment, we're getting about 35% cleavage efficiency. So these are, as far as what I've seen, these are currently the best cleavage efficiencies I've seen in iPS cells with any CRISPR-Cas or any genome editing tool. So briefly, we wanted to look at different cell lines with our different tools, and now we've evolved a little bit past the efficiencies of our total all-in-one vector. So here we're looking at 3 different cell types, and the different columns represent either our vector system. The red column is our Cas9 mRNA and U6 guide RNA, and the green column is our total RNA solution. As you can see in every case, the mRNA works significantly better or the same as the plasmid solution, but the total RNA solution works significantly better in any case than any of the other solutions. So just an extra couple hours of doing an IVT reaction, purifying it, you generally get significantly more, at least in this case so far, significantly better efficiency of cleavage. The other benefit that brings is having an mRNA solution. Basically, there's some chance of getting the gene integrated into your genome, so that there wouldn't be any chronic expression of your editing tool, and we get a much earlier onset of peak activity. So there's a burst of translation that occurs. The translation of the protein occurs at the same time that the guide RNA is there, and the RNA is not degraded yet, so we get a much higher efficiency. One thing that we and others are working on right now currently is that we're looking to replace the Cas9 mRNA with actually a purified Cas9 protein. And what we're seeing is that even gives us a more efficient reaction and with even an earlier onset peak activity of maybe 12 to 24 hours. So another application side, we were interested in engineering different cells in the applied space, very focused on CHO cells. And this is some data I just got this week, actually, when we looked at CHO cells with our new delivery format. This is our new messenger max lipofectamine along with our total RNA solution. And now we can see rather remarkable cleavage efficiencies of over 50%. And generally working in CHO, we were very happy with 10% at this locus. And now we're well up there in 50% range. So this is very encouraging for the types of engineering that we want to do in these cells as well. Finally, we want to multiplex. So we're looking at multiplexing again. This is some very fresh data. Again, we're comparing against our vector format in the purple, and then we have different combinations of guide RNAs. And if you focus on column 4 and column 9, those are 3 different guide RNAs, and these are both taken from the same pellet. And we can show that we're getting good cleavage at all these loci that were tested in the same cell. And we're in the process of cloning out these cells from this population to then score how many of these clones actually have all edits in the same cell. And we expect that to be a high number. And then I'll be reporting on that next time. So finally, one slide on our library kit. So I mentioned that we can deliver Cas9 in a lentivirus particle for doing a high-throughput screen. What that allows us to do is to have a particle with one Cas9 and guide RNA sequence in it that will target a single gene. We can set up our transduction to be able to have an average of 1 particle hit per cell in the pool, and select that such that we have a pool of cells that on average have a single gene knockout. And then that allows us to do a lot of different studies. We can do survival screen. We can do these experiments, these knockout experiments in some of our reporter cell lines. For instance, we have reporter cell lines that have a fluorescent reporter that indicates activity of a particular kinase cascade. So now we can do knockout screens in the presence and absence of maybe small molecule drugs and overlay that with activity of a particular kinase cascade. And now we can get some idea of what the cross-reactivity of different proteins would be when they lose their function to this cascade. So I believe that we're going to see a lot more of this type of screening in the field, potentially at the expense of doing shRNA, siRNA screening. So that's my prediction for the future. So finally, this is just to wrap up. We're looking at Cas9 as transformative technology. In the literature, you're seeing more and more papers every day. Our goal is to make this workflow easier and make it such that the researcher does not have to be a genome engineer to do this, to allow the researcher to easily engineer their cell and get to the important experiment that they're interested in. And that's not only in creating cell lines, but also creating small animal models. So finally, that looks like all the tools that we have in the workflow. So you can go through our catalog online or talk to your local sales professional, and they will help you work you through the entire workflow of reagents that have been shown to be optimized for you doing these gene editing exercises. Everything I've spoken about today, we do have a services group in Carlsbad that will do these for customers. So if there's a customer that is either unable or unwilling to do this type of work in their lab, we're happy to discuss with you the potential for doing that work for you. So creating knockout and knock-in cell lines, we're seeing a lot of interest and excitement in that. And we do make CRISPRs and TAUs and validate them for customers. We do produce lentivirus. We will be producing lentiviral libraries. Those CRISPR libraries, our first libraries will be coming out probably in October and November time, followed by a lot of other different configurations, and also other standard molecular biology procedures like sample prep and cloning and cDNA library production. So with that, I'm going to end up. Happy to take any questions. I hope this was informative. Thank you.